This recipe details how to overlap significant copy number variation regions with known gene annotations fetched from UCSC, and then extracting the gene list that the CNV regions lie in. In this case, we identified these CNV regions from SNP array data using the GISTIC algorithm within GenePartner. We then perform Go enrichment with the list of genes using MSigDB. This allows for the identification of potential biological functions associated with the CNV regions. We begin by creating a subdirectory in our home folder called CNV function. We will now fetch RefSeq gene annotations from the UCSC table browser. In the UCSC table browser, we now set a couple parameters. We begin with clade primates, genome human, assembly HG19, genes and gene prediction tracks, track RefSeq genes, table refgene, and we want to go for the entire genome, and we want bed format output. We want to send it to GenoSpace and name it HG19 refseq.bed. Now hit get output. Here we select whole gene and hit get bed. This will send the bed file to GenoSpace. We now move the annotation file from the GenoSpace home directory to our subdirectory by dragging and dropping. As you can see here, it is back into the subdirectory. We now select the annotation file and send it to Galaxy. This will import the annotation file into Galaxy. We now also want to get the file of CNV regions generated from GISTIC. We go to Get Data, GenoSpace Import, and go to Public, Share Data, Demos, Analysis Recipe 3, GISTIC regions.bed, and hit Send to Galaxy. This will import the CNV regions file into Galaxy. Now I want to overlap our regions with our annotation file by going to Operate on Genomic Intervals and then clicking Intersect. Here we select overlapping intervals of our refseq annotation file with our GISTIC regions. Select overlap of 1 and then hit execute. Now that the job is done, we can see that the fourth column contains the refseq IDs of the genes which overlapped with our CNV regions. To extract them, we go to Text Manipulation and hit Cut. Here we say Cut column C4, delimited by tab from Intersect Output, and hit Execute. After the job is done, we want to export the list of refseq IDs to GenoSpace by clicking Send Data to GenoSpace Exporter. We select the cut file and browse to the CNV function subdirectory. We name it gistic_genes.txt and hit execute. After a brief period, this will send the cut file back to GenoSpace. Now, back in GenoSpace, we can see that the cut file is in our subdirectory, as seen here. It only contains the refseq IDs. We want to send this list of genes to MSigDB to check for significant biological function overlap. In MSigDB, it will automatically populate the list of gene identifiers. Here we want to select C1, C2, and C3 and select refseq IDs. We now compute overlaps. This will now show a list of significant overlaps if found. We now want to export this list to GenoSpace by hitting Send to GenoSpace and then selecting the proper subdirectory by clicking CNV functions and then hitting Submit. We can now hit close and go back to GenoSpace. Here we can see that the overlaps.txt file is within GenoSpace. If we hit preview, we can see that it is a simple text representation of what gene sets significantly overlap with the gene set we found to lie within our CNV regions. Thank you for watching, and please stay tuned for more recipes.